So let us study about these very important cells here now, which we are going to study here are NK cells, natural killer cells. See, the name itself tell you that they are natural killers. Natural killers like Naxalites. The name is Naxalite cells, natural killer cells. They are born killers. They are born killers. So they don't require any training or presentation. So they are perfectly born killers. They don't require any training. So why don't require any training? Because they are a part of your innate immunity. They are a part of your innate immunity. And remember, because they are natural killers, okay, so they are not kings like T cells. So kings have their own tantrums. They don't have their own tantrums. They have no tantrums. So they do not require presentation of antigen by MHC complex. They do not require any presentation by MHC complex. So can I say NK cells are not MHC restricted. They are not MHC restricted. They don't require MHC complex. They can identify the problem and they can kill that there and there itself. So they are natural killer cells. The other killers were CD8 cells, okay, cytotoxic killer cells. They are natural. These are natural killer cells. So let us see how do they look like. So natural killer cells actually look like large granular lymphocytes. They actually look like large granular lymphocytes. So if you look at them, they usually have granules in the cytoplasm and they are little larger than the lymphocytes. So they are larger than the lymphocytes and they have granules in them. So they are called large granular lymphocytes. That's the typical picture of it. And these NK cells constitute only 5 to 10% of all lymphoid cells. So they are very few in number, only 5 to 10%, but they look like large granular lymphocytes. So if they look like large granular lymphocytes, what is the property? Their property is that they can stick to any cell. So they are among common people, they are among the commons, they are like common people and they can stick to the other cells. So they have a very powerful sticking mechanism, okay. This is called natural killer cell adhesion molecule. It can adhere to any cell and for this it has a marker called as CD56. So CD56 is called as natural killer cell adhesion molecule. So CD56 is a CD marker for identifying the cell adhesion molecule present on the NK cell. So any NK cell adhesion molecule can be identified by which CD marker? CD56. Now let us see why I am saying you that they are one among the common and they do not require any antigen presentation. So look at this, how NK cells identify the pathogen or any problem. See, any NK cell has two arms, okay, it has two arms, isn't it? So everybody has two arms, everybody has two hands only, okay. So they have an activating arm, okay, they have an activating arm and they have an inhibitory arm. So they have an inhibitory arm and they have an activatory arm, okay. So activating arm is also called as NKG2D. Okay, activating arm is also called as NKG2D. Now, let us see what happens, okay. So normally the cells are able to move over every cell. It keeps on checking every cell, okay. It keeps on checking every cell. So normally, normal circumstances, the inhibitory arm of the NK cell binds with the MHC1, which is present in all the nucleated cells of the body. So all the self cells of the body, all the nucleated self cells of the body have MHC1. Okay, so what happens is inhibitory receptor of the NK cell normally binds with MHC1 molecule of the self cells. And when this happens, this cell is inhibitory. This cell means everything is okay. He gets that nirvana, moksha, everything is okay, no problem. So he's very happy, okay. So he keeps on going on. He keeps on checking every cell, okay, everything is okay. Now, MHC1 is a big molecule. It has lots of HLA, isn't it? So out of all the HLA, which is the typical HLA molecule which is expressed by MHC1? Remember, it is HLA-E. Okay, so remember HLA-E is the self-recognition target of the NK cells. So which is the self-recognition target of NK cell? It is HLA-E. Okay, so out of so big MHC1 chain, which is the self-recognition target of MHC1? It is HLA-E. So it checks HLA-E. So that is why they can ask you, what is the self-recognition target of NK cell? So self-recognition target of NK cell is HLA-E, which everybody should remember. Now, of full MHC molecule, only HLA-E is the self-recognition target. Now, let us say, suppose a virus, I'm going to draw this, a virus comes into the cell or cancer starts developing in the cell. So let's say a virus has entered into the cell or cancer starts developing in the cell. So it is postulated that when the viruses or cancer enter into the cell, the MHC1 expression of the cell decreases. The MHC1 expression of the cell decreases. Okay, so what happens? When the MHC expression of the cell decreases, the inhibitory arm does not have something to bind to. Inhibitory arm does not have something to bind to. So as soon as the inhibitory arm doesn't bind, the activating arm will get activated. The activating arm will get activated and it will kill this cell. Okay, it will kill this cell. And how does it kill? It kills by either fast ligand pathway, or by perforin granzyme pathway, So it either kills by fast ligand pathway or it kills by perforin granzyme pathway. So as soon as the virus or cancer enters into the body, the MHC expression decreases. So the activating arm of the MHC1 gets activated and kills the cell either by fast ligand pathway or perforin granzyme pathway. Now, here is the catch. Okay, so here is the very important catch point that you have to remember. Suppose I am saying, that we have already read this killing mechanism. If I am saying we have already read this killing mechanism, is it true? Yes, you studied that cytotoxic T cell, that is CD8 T cells, also kill by the same mechanism, by perforin granzyme pathway or fast ligand pathway. The same way they kill. Okay, so they kill by either fast ligand pathway or perforin granzyme pathway. Okay, so the same pathway of killing, isn't it? So how are they different from CD8 cells? So let me explain it to you with an example. Okay, so if you understand this example, you will understand this well. So let's say this is you, okay, and you are stressed. You do not know that you are stressed, but you are stressed. Okay, so what happens is your friend, very close friend, comes and say, you look stressed today. You say, no, I am not stressed. You say, no. He says, no, you look stressed today. Let's go out. Let's have some party and come. And he acts immediately and your stress is relieved. Okay. So who is this friend? This is NK cell. Okay. But now you know that you are stressed. Okay. You know that you are stressed and you go to the psychiatrist and tell him that I am stressed. And then psychiatrist treats you. So what is psychiatrist? CD8 T cell. Do you understand the difference? See, when the virus enters the body, this cell itself does not know that I am infected. But MHC expression decreases. So who identifies this? NK cells. That means this cell is stressed. He himself doesn't know this, but who identifies it? NK cells. So can I say NK cells help in doing stress surveillance? NK cells help in doing stress surveillance. Now, let's see the another phenomena. So now this cell identifies that I am infected by virus. Then they give this virus to their MHC molecule, MHC1. This MHC1 takes it to CD8 cell and then CD8 cell kills it. So this CD8 is the psychiatrist. This person takes the problem to CD8 and then he acts. So that is late, isn't it? That's CD8 cells. So remember, CD8 is also a killer cell, cytotoxic killer cell. NK cell is also a killer cell, natural killer cell. But what is the difference? MHC presentation is not required by NK cell. NK cell does not require that antigen is presented to MHC1 and then MHC1 gives the antigen to NK cell. No, it is a stress surveillance cell. As soon as some change occurs to the NK cell catches it and he says something is wrong. And he doesn't wait, he kills that cell immediately by activating the activating receptor. You understand? NKG2D. It kills immediately. So this is your natural killer cell which everybody should be knowing. Okay. So he is a stress surveillance cell. It is a stress surveillance cell. So that is how it acts. Everybody clear with this? That is how the NK cells act. Now, once you're clear with this, let's go to the next phenomena. Okay. What is any other way in which NK cells can identify the problem? Yes. See, whenever the cells are coated by antibodies, whenever some cells are coated by antibodies, especially IgG antibodies, do you know IgG antibodies are incomplete antibodies? So these IgG antibodies can coat the cells, but they cannot lyse it. So such cells are called sensitized cell. You will be studying more about it in blood transfusion. These cells are called sensitized cells. Okay, why doesn't it lyse it? Because IgG antibody has small arms. So it cannot overcome this zeta potential which keeps the cell separate. So therefore, it can just coat it and it cannot lyse it. Such IgG coated cells, if it is of hematopoietic lineage, are identified by the spleen and spleen destroys them. Okay. But what about the other cells? Who identifies these antibody coated cells? So these antibodies coated cells can be identified by NK cells. These can be identified by NK cells. So remember, NK cells have a special molecule on them. This is called CD16. NK cells have a special molecule on them called CD16, which can identify the Fc receptors of IgG. Okay, which can identify the Fc receptors of IgG. Okay, so immediately NK cell, if it identifies that the cells are coated by IgG, it immediately kills them. So can I say that NK cells kills antibody coated cells? So what is this mechanism called? Antibody dependent cellular cytotoxicity. What is this called? Antibody dependent cellular cytotoxicity. So NK cells are able to kill any cell coated by antibody. So this is called as antibody dependent cellular cytotoxicity. That means, look at the name itself. Name itself is telling you so much. Antibody dependent, that is those cells which were coated by antibody, they were killed. Cellular cytotoxicity, those cells were killed. And who kills them? NK cell. And if you go to your hypersensitivity lectures, we have already discussed about it, that this is called type 2 hypersensitivity. So whenever killing is by ADCC, this comes as a part of type 2 hypersensitivity reaction. This is type 2 hypersensitivity reaction. So everybody clear? So can I say there are two markers which I can use to identify NK cells? One, I have already studied that NK cells have a cell adhesion molecule, that is CD56. Other, I studied that NK cells have a special marker called CD16 by which identifies Fc coated cells. So can I say that any NK cell can be identified with the help of two CD markers, CD16 and CD56. So these are the two molecules which can be used to identify NK cells. These are the two molecules which can be used to identify NK cells. Are we clear about NK cells? So what did you learn? NK cells are not MHC restricted. Okay, so they can be identified by 16 and 56. ADCC is a mechanism. Stress surveillance is a mechanism of MHC molecules, of NK cells. Now once we are clear with this, now come to next thing. Next very important point that you have to understand is that NK cells can also produce cytokines and they can also respond to certain cytokines. So they can produce cytokines and they can also respond to certain cytokines. So remember always that NK cells, okay, can release interferon gamma and this interferon gamma activates the macrophages, and these activated macrophages then can kill intracellular pathogens. Okay, so remember this pathway. This pathway of releasing of interferon gamma was actually a function of T helper 1 cells, which we have also talked. But NK cells can also release interferon gamma to activate the macrophage for killing the intracellular pathogens. So it is helping macrophages to kill, okay, the intracellular pathogen. So it's a part of your innate immunity. Okay, it's a part of your innate immunity. Now, so that is, it can release cytokines. Perfect. Now, which are the cytokines which stimulated? See, NK cells first can be stimulated by interleukin 12. Remember, macrophages itself produce interleukin 12, which stimulate NK cells to release interferon gamma, so that this interferon gamma activates the macrophage into activated macrophages, and then they can kill the intracellular pathogen. So interleukin 12 supports the release of interferon gamma. So macrophages themselves stimulate NK cells for killing. Okay, so they are good friends. Macrophages or NK cells are good friends. Apart from them, remember, NK cells are stimulated by two more interleukins, interleukin 2 and interleukin 15. Okay, 2 and 15. So remember, interleukin 15 you will be studying later when you study immunodeficiency diseases, that interleukin 15, interleukin 2 are very important interleukins which come from the gamma receptor of the cytokine. Okay, so this, whenever interleukin 2 and interleukin 15 are absent, this usually occurs in SCID, okay, severe combined immunodeficiency. Then in especially X-linked, okay, X-linked recessive type, then in this NK cells are also not produced. That is why these patients have not only absence of T cells, they don't have NK cells also, because there is absence of interleukin 2 and interleukin 15. So they suffer from this disease. Okay, so this you will be studying in immunodeficiency. So remember, 2 and 15 stimulates NK cell. Now apart from that, you should remember interleukin 2 is a very special interleukin. So interleukin 2 is actually produced by T helper 1 cells, whose release interleukin 2? T helper 1 cells release interleukin 2, and this interleukin 2 has three major functions. One, just now you studied, it activates NK cells. Second, it stimulates Th1 cells by paracrine loop. More Th1 is produced. And third, it can activate regulatory T cells. So regulatory T cells have a special receptor on them. So if you look at the regulatory T cells have a special receptor on them. This receptor is interleukin 2 receptor. Okay, through which regulatory T cells can also be stimulated by interleukin 2. And I hope you remember, this receptor was used to identify regulatory T cells. So CD25 is a CD marker to identify interleukin 2 receptors. So therefore, CD25 is a marker to identify regulatory T cells. So if we talk about, so we have talked about regulatory T cells before. So everybody please go back and see T cells and see what are regulatory T cells. They suppress the immunity. So can I say interleukin 2 is a very important interleukin. It does three functions. It can activate NK cells, it can activate regulatory T cells, and it can also activate Th1 cells. So this was interleukin 2, which you should remember. So what are the three interleukins secreted by which stimulate NK cells? Interleukin 15, interleukin 12 and interleukin 2. So interleukin 12 is the one which helps in releasing interferon gamma from the NK cells, which everybody should be clear. Now, everybody clear with this? Okay, so once you're clear with this, now let's come to the very important updates that have come up recently, and everybody should be knowing these updates because these are the things which they ask you in exam. So these are very important updates which everybody should know. So there are two terms, okay. So first term is called innate lymphoid cells. Okay. So remember, what is innate lymphoid cells? So they found certain lymphocytes, which behave like Th1 cells, but they do not have T cell receptors. So some lymphocytes which behave like T helper 1 cells but do not have T cell receptors. So these molecules can release molecules like Th1, Th2 and Th17. Okay, so they are a type of, basically they can release, they can function. Okay, so they are the type of T lymphocytes, especially helper T lymphocytes, which can produce cytokines similar to Th1 was the first one that they identified. Then they said no, they can also release Th2 like cytokines or Th17 like cytokines, but they don't have T cell receptors. So they are not T cells actually, but they behave like T cells. So they can release interferon gamma. They can also release interleukin 4 and interleukin 5. They can also release interleukin 22. So these cells, okay, behave like T cells, but they don't have T cell receptors. So they said NK cells could be a type of innate lymphoid cells, because you now know NK cells do not have T cell receptors and they can release interferon gamma. So can I say NK cells were the first type of innate lymphoid cells to be identified? So which were the first group of innate lymphoid cells that were identified? It was NK cells, which were first group of innate lymphoid cells which were identified. So that's the first update that you should know about innate lymphoid cells. Now let's come to the second update, okay, which recently came up. Okay, these cells were called NK T cells. So these updates are very important for your central exam. So please, everybody should know that. NK T cells. So what do you understand with the term NK T cells? So these are the T cells. Okay, so they are the T cells which have markers of NK cells, which can express markers of NK cells. So they are actually T cells. Okay, so whatever you write behind is the type of cell, T cells. They are T cells with markers of NK cells. Okay, so they are T cells, but they express markers of NK cells. So they identified these molecules and they said, what are they? So then they found out that they are very special molecules because these type of molecules can respond to glycolipids. They can respond to glycolipids which are presented by MHC like molecules. Okay, which are presented by MHC like molecules, especially by CD1. Okay, especially by CD1. So they identify that NK T cells can identify the glycolipids which are presented by MHC like molecules which have CD1 on them. So what is this MHC like molecule? So these MHC like molecules were identified by CD1. So they introduced a term that they are CD1 restricted. They are CD1 restricted. So these NK T cells were CD1 restricted. So very important update, please everybody to know this. Okay, so CD1 restricted cells, which are CD1 restricted immune cells, they are NK T cells. Okay, so they are NK T cells and lots of research is going on these cells. Okay, because they typically identify glycolipids which are presented by MHC like molecules, especially which are CD1 restricted. So these molecules are NK T cells and yet lot of research is going on that. So everybody should be clear with these two updated terms which you should know. Innate lymphoid cells. So innate lymphoid cells are lymphoid cells with no T cell receptors, but they respond like CD4 cells because they release cytokines like that. So NK cells were a first group of innate lymphoid cells and more research is going on this. Second is NK T cells, which they found out that they are T cells with NK cell receptors and they are CD1 restricted and they can respond to glycolipids responded by MHC like molecules. Everybody clear with this? Yes. So that is the update. Now let's see one image which I am going to show you. You need to identify these cells. So look at these cells now. Can you see? So the nucleus is little larger than the size of small RBC and it has lot of granules in them. So little larger with granules. So this is a large granular lymphocyte. So NK cells belong to this group of cells. Okay, so these are NK cells which belong to large granular lymphocytes. Okay, apart from that, one more just last point about NK cell. So in hematology we will be studying about a lymphoma which is a very unique lymphoma And just last point about this, so that will complete this, NK T cell lymphoma. So I'll just give you 2 points regarding this which you should remember. See, name is NK, okay, starts with M, so typically affects nose, so they usually has septal perforation, okay, okay. So these patients usually present with septal perforation, okay, and these are very fatal, okay. So these people who get this lymphomas usually die. So earlier days they were not able to identify this, so this term was misnamed as lethal midline granuloma, okay. So it was misnamed as lethal midline granuloma. So please remember, lethal midline granuloma is the other name for NK T cell lymphoma. Lethal midline granuloma is the other name for NK T cell lymphoma. This is lethal midline granuloma. So it has septal perforation, 1. Number 2, you have to identify that these are usually associated with Epstein-Barr virus infection. So normally Epstein-Barr virus typically causes B cell neoplasms, but it can also cause NK cell neoplasm, and this is NK T cell lymphoma. So these are 2 important points about this lymphoma which I want you to identify. So they usually cause septal perforation, so it is also called as lethal midline granuloma, and they usually associated with Epstein-Barr virus infection, okay. So with that we'll be finishing about NK cells.